So in the sake of time, we'll just get straight into the talk. So just as a brief overview, I'm going to talk about how my view on using databases and integrating that information into hypothesis generation came about. And really it's from my first postdoctoral work, which I'll outline in some detail. We'll then talk about what I like to call the data tsunami. So we have this logarithmic increase in data that's being produced by many different types of biological projects, whether it's environmental data, whether it's metagenomics now being a buzzword, whether it's traditional genomics or organism sequencing. And it's starting to outstrip our ability to organize, to search, and to analyze this data. But certainly there are data sets that we can access now as basic scientists with some help from our bioinformatician friends, and that'll be one of my take-home messages at the end. So we'll look at some of the databases that are out there and why we should be using them for particular parts of our project or for testing hypotheses. We'll then look at how my laboratory, in collaboration with Dr. Jason Berman at the WK Health Center, have been using databases and these different gene data sets to try to develop hypotheses and approaches to looking at T-cell leukemia. So how did a wet lab scientist like myself get interested in using databases? And this would date me, I guess, back in the 90s when I actually had some hair. And I put on purpose one of these IBM clones, which unfortunately was the tool of the time in the 90s. And for those of you who are old enough to remember, we started off with these very large 8 or 9 inch disks to 3.5 disks. And then if you're really lucky, in the late 90s, you could have a zip disk. And that was maybe a few hundred kilobytes of data. And my entire PhD thesis and all the data fit on one zip disk. And that's what I carried with myself from Montreal to Scotland. So a long, long time ago in a place far away, this is a beautiful shot of Edinburgh, Scotland, with Arthur Seat in the background, which is actually a caldera from an extinct volcano. And I worked at the MRC Human Genetics Unit, as you heard from Joanne. And I worked with someone named Professor Wendy Bickmore. And the claim to fame for the Human Genetics Unit there is not just their work on genetics, but their really early work on epigenetics, on the organization of chromosomes. And in fact, that was Wendy's main research area. And so what she started off with was asking me to clone and characterize some novel nuclear proteins. So she's interested in the organization of the nucleus. And what she explained to me seemed really simple, and I think I was quite naive to take on such a huge project. But what she had done is taken a retrovirus that carried an enzyme that both conferred resistance to an antibiotic, but also, when you added the right substrate, would turn the cells blue. And this vector would integrate into genes randomly in the genome, but it had a splice acceptor, so it would splice in with the endogenous sequences in the genome and thereby trap, so it's called gene trapping, trapping those sequences. So you'd know nothing about them other than we would try to map where the localization was. But by the fact that we had this enzyme now linked in frame with that protein, you'd either get blue cytoplasm or you get blue nuclei, or sometimes both. And so we took all the cells with blue nuclei, single cell cloned them, and then started the process, the laborious process of cloning individual genes. And I was given the secret project of these two gene traps, codenamed CT143 and CT146. And this is at a time where when you tried to search databases, everything was unknown. You literally had no information to determine what that would be. So by the localization, we knew it made spots. Those spots were in the nucleus. They looked kind of similar to each other, yet, as I'll show you later, they're profoundly different in their function. So back in 1999, this was really laborious, trying to clone genes. It was many screening steps, lots of radioactivity. In fact, most of the students in this room, if they worked with radioactivity, it would be a very minor amount, and probably most of you have never used any radioactivity. We were using industrial amounts of radioactive phosphorus. The Human Genome Project was just wrapping up, so there were sequences available, but there was no scaffold. It was really poorly annotated. There were whole chromosomes missing from the initial sequence that was available. It was not easily searchable. There were no gateways to search it. However, there were databases available with what were called expression sequence tags. So this is where they created really short reads of transcript data. So there were cDNAs And they spanned many thousands of genes across many organisms. And so you could start looking at those sequences. And I purposely picked the circa 1999 logo from NCBI, so the National Center for Biotechnology Information, just again to show you that this is kind of been a stalwart of informatics for many basic scientists. You're probably most familiar with NCBI using PubMed, which has now become the kind of repository for most scientific literature. And we were using this thing at the time which we thought was fantastic, which was GenMake, which has now been pretty much hidden within NCBI and the huge amount of informatics that's located there. And we had this thing called Basic Local Alignment Search Tool. And just like Google now is, the term Googling is used to search something on the web, you may not, for the students in the room, you may not realize that this is where blast came from. So when you're blasting something against a database, you're actually referring to this tool that was developed in the early 90s. And in fact, that was the major tool that we had. So we would blast our sequences against these EST databases. And so we're lucky that as the human genome was wrapping up, the technology was applied to smaller genomes because they could sequence much more rapidly. And the Sanger Institute in particular was one of the major innovators in this area. So they were busy sequencing worms, flies, and yeast. A lot of the fly work was actually done in California. And those genomes were actually available many years before the human genome sequence was available. And so we would search those databases and look for similar sequences. Now, a particular quirk of evolutionary biology is that there's much more difference, possible differences between the nucleotide sequences of a paralog protein in two organisms than there is at the protein level. So it's harder to change the amino acid sequence without affecting function, but because of different codon usage in different organisms, you can really change the nucleotide sequence. So I would convert my cDNA sequences I had into an amino acid sequence, and then I would use those. This would be commonly called like a T-blast, for example, would be the type of search. And effectively what we were doing was comparing those sequences to the protein domains between species. And this is really the beginning of comparative genomics as a field, where individual researchers like myself who didn't have access to mouse or human genomes that were well annotated, but could go, and certainly the yeast genomes were the first to be very well annotated, and then eventually Drosophila. You could then compare between organisms and try to get an understanding of function. You could use also databases and tools like the Simple Modular Architecture Research Tool, and I still really like this tool, even though there's been something similar that's integrated into NCBI. And what you could do here is insert your sequence and it would give you back a cartoon. And this cartoon would tell you, well, there's a domain here called an SR domain, so it stands for serine arginine. And this is often found in splicing proteins. And then you knew it had a kinase. So now I knew that CT143 was a kinase. Excellent. And it had a domain found in it that might be involved in splicing. So now, without any other information, I had a hypothesis that this is a splicing protein that was a kinase that might function in alternative splicing genes. Then through our comparative analysis, I got hit with this protein, the pre-RNA splicing factor 4, which in Schizosaccharomyces pombe is actually a protein kinase involved in affecting splicing specifically. So now I had a defined biological role for a similar sequence in another organism. And for many of you, if you're working on proteins that are not well defined, I would keep checking these other organisms. That's a really important point. You need to be doing comparative genomics if you don't have a function for the protein you're working on. So now we had CT143, we knew it was a kinase. We knew that it was similar to PRP4, so we called it PRP4 kinase. That seems simple, right? Easy to remember. Well, HUGO, which is the Human Genome Organization, which is responsible for nomenclature, doesn't really care much for your feelings, or history, or any previous nomenclature. They just want a different name from everything else in the database. So, what I learned since 2001 is that pretty much every protein that is named, unless it was an extremely unique name, would be altered later. So we were able to publish this work in Molecular and Cellular Biology. We then found what CT146 was. It turned out to be a chromatin remodeling factor, well known in Drosophila as ACF1, which then Hugo said, well, BAS1A, that's a good name, it's different. No relationship to any of the literature, comparative genomics that was used to discover the function of that protein. And the reason why I'm illustrating this right now is we're going to talk about some tools like String, which I'm going to introduce in a little bit. And those databases use automatic annotation from abstracts. or text searches. This is going to cross-link all your data sets. So you're going to have information that's included in those databases as being actual information associated with that gene, but it's associated with the wrong name. And that's going to affect a lot of your analyses. So you really have to be aware of that and double check. So sometimes when something seems too good to be true, it could be, and you want to double check that. So this work was published on ACF1 in EMBO. So whereas this was a splicing kinase, this turned out to be a chromatin remodeling factor that's actually quite essential, both in DNA repair and in replication of DNA. And just as an example of, there's no such thing as bad data, and data never really goes away. Everyone on the planet, pretty much, that is working on ACF1 is using my cDNA that I cloned as a postdoc. And fast forward from 2002 to 2013, and I'm working again on ACF1 with Aaron Godarzi at the University of Calgary, supplying once again that cDNA and looking at its role in DNA repair. Did I mention we were working on 1,200 of these gene traps? So I had 2 that I had to personally characterize, but there were over 1,200 that we had to deal with. And when I started in 1999 in Wendy's lab, that's what it looked like, times 1,200 binders. We literally had a room full of paper. We decided, well, this wasn't very efficient. We should probably put it into a computer somehow so we could efficiently access that data. And what that resulted in was the creation of the nuclear protein database, which was a project between myself and my wife, Rachel Farrell, who is a database specialist and a bioinformatician. And so now, today, something else I'll point out is that this database is 12 years old, I think, and it's still functioning. It still works. This is a rarity for most web-based tools. Their half-life in general is 5 years, which coincidentally is about the length of a PhD. Okay? So these projects are provided by PhD scientists. They're worked on, they leave, and then they die. And they die a horrible death. And in fact, NCBI, at one point that we all use, nearly died a similar horrible death. Okay? So it was actually people in Congress who were arguing it should be shut down. And it's still going, thanks to people like Harold Varmus and various other people who are involved in realizing that these were important resources and they should be maintained. Now, when it comes to individual tools from small academic labs, we don't have that kind of horsepower behind us to keep these databases going. Luckily, Wendy Bickmore believes in the nuclear protein database, and we've kept it going. So this was published in Nucleic Acid Research. I put this on here to say that if you don't know what tools are available, every January, Nucleic Acid Research publishes a database issue, and it has all the latest databases, and including updates of very popular databases. So it tells you about the tool, what they can be used for, gives you the URL, so you can find those databases. So if you never pick up Nucleic Acid Research, that's a reason to pick it up, and certainly the January issue you'll find really useful. On to the second part here, part 2 of the talk, talking about this data tsunami, and this is actually an image taken for a cover of a book that myself and Jason Berman are editors for. That should be coming out. It's available for pre-order from Amazon on Cancer Genomics. And what it is to illustrate that we've got this large body of data that is going to crush us if we don't start using it and start developing ways to analyze this information. And it's all about sequencing. And what's happened is that what we call traditional Sanger sequencing, so chemical sequencing, was used for over 25 years. And it was a good process, but it wasn't very scalable. And what's happened now is we've gone from a snail's pace in using basically Sanger sequencing approaches that took 11 years and $3 billion to do one human genome, which Cellera then through shotgun sequencing. So here they were actually walking along large contigs of DNA that were in BACs or YACs and sequencing them and walking along from primer to primer. Whereas Cellera was just sequencing everything at random, but this was not possible without a scaffold, okay? So that's a take-home message here, that shotgun approaches don't work without a decent scaffold or these longer contigs of sequence so that you can anchor where the position of those reads are. Now we're at moving from snail to a turtle to a rabbit's pace, where with next generation sequencing, and this number is completely wrong now, it was in 2012 11 days and $5,000 to $10,000 per genome. This is now down to probably 5 or 7 days. I think Dan Gaston has more modern numbers here in the audience. And they're trying to approach $1,000 per genome. And what had happened was with Sanger sequencing, before really the widespread use of of next generation sequencing, it was following inverse of Moore's law. So it would be, the cost of a genome would be half what it was every 24 months. Until we had these next generation sequencing technologies available, and then the costs were really driven down. And it's nice to see there isn't one flavor of next generation sequencing, which creates some competition, which also keeps the cost of the infrastructure down for us. We're going to switch from looking at DNA sequence to looking at transcriptome sequence, or the sequence of messenger RNAs. And I want to point out that there are a lot of freely available databases and gene sets that you can look at. A lot of this data, since I would say 2001, was from expression arrays, and now we're moving into RNA sequencing, which since 2012 has really started to take over for transcriptome projects, certainly for discovery research. I would still argue that it's more cost effective if you have a known gene set to use gene arrays. But if you've got a new cell line or a new condition, RNA sequencing is much better. What this does is that you get a lot of short reads from a messenger RNA, which would then use the scaffold of the human genome to identify, for example, splice junctions. You can also identify non-coding RNAs and microRNAs, which is something you can't do with gene arrays in a single experiment. You have to actually choose those sequences and then interrogate them. So if you're doing a more open project where you don't know what you're looking for, RNA sequencing is probably a much better approach, and the costs of this are continually dropping. So where do we start? Well, one of the great places to start, and I've already mentioned them several times, is NCBI. And you can go to geodatasets. And I would suggest that this type of data set is interesting for general transcriptome data. So if we go back to our favorite protein here, or our favorite gene, PRP factor 4B, you can type in that. It'll give you a bunch of different experiments that are sitting in the database. And here we have one with endothelial progenitor cells from cord blood versus adult peripheral blood. And we see a difference in gene expression. And this is a single replicate from a data set, usually of 3 or more replicates. Just for your students, you can kind of say, well, that looks interesting. You can visually scan. It can be rank ordered by fold change or by p-value. And there also are tools that will let you analyze the entire data set or compare data sets. So that's something that I think the students can get working on right away. And it helps you develop hypotheses of what pathways that the particular gene you're interested in is working on. Now, if you're more interested in cancer genome data, then I think the Cancer Genome Atlas data portal is a fantastic resource. And in fact, this is something that I want to spend more time using myself for my own research. And it's really good for cancer-specific sequencing and transcriptome data. It has everything from Affymetrix style gene expression data to exome arrays to microRNA profiling to RNA-seq, so you get some splicing information as well. I would say that this is less simple for a non-bioinformatician to access. I think you really have to ask a bioinformatician to help you with these data sets. There's less kind of pre-crunched analysis available, although, and some of the data sets are still protected as those publications come out. Another level, so rather than just looking at the transcriptome and looking at the DNA sequence, is trying to integrate all the information that's involved in regulation of genes. And that's what the Encyclopedia of DNA Elements is about, or ENCODE project. And so it's an international collaboration of research groups that are funded by the National Human Genome Research Institute. And their goal is really to provide you a comprehensive parts list of all the functional components of the genome. So that includes not just the transcripts and the splicing data, but also how the DNA is organized in the nucleus, whether they're hypersensitivity sites, the chromatin that packages that DNA, how it's modified, how it's organized in the genome, and what factors might bind it. This is the typical view that you might see if you use a genome browser, and there are a couple different flavors of genome browsers. So I prefer the one at University of California, Santa Cruz, but there's also a great one in Europe from Ensemble, which allows you to view tracks of transcriptome data mapped against the human scaffold, tells you the chromosomal position, tells you what histone modifications there are. You can add your own personal tracks of your own data and analyze what information you might have. Like, let's say you have some of your own chromatin immune precipitation data. You can overlap that with the scaffold and include all this other information. And we find this is, again, really good. For example, if you found a gene and you say, well, what regulates it? And then you happen to see, oh, this transcription factor binds here. So for example, you might see a CBCF1 binding site, which is a transcription factor that works with Notch, which we're going to talk about a little bit later in my talk. you might predict that that gene is regulated by the Notch pathway. So this is the type of data you can get and directly download from ENCODE. I wanted to include this because I've found in talking to these kind of audiences before that the students found this the most useful type of database. So there are over 700 commonly used cell lines that are included in this database from the Broad. So it's called the Cancer Cell Line Encyclopedia. You can get a free account. Once you're registered, you have access to this data. And what it does is it provides you the normalized transcriptome data and copy number variation for every gene in the human genome. And what you can do is it'll automatically rank order them from cancers that are clustering with low expression or high expression. And here we're looking at the cadherin 1 gene, which is a tumor suppressor that's lost in aggressive cancer and promotes metastasis. And what I look for in someone who's looking for biological markers that will stratify patients for differential response or differential outcome is to look for a large variation. That means within one cancer, there are cancers that have low levels of cadherin or high levels of cadherin. And so you can expect that one would either be maybe more metastatic if it had low levels of cadherin and less metastatic. And then you have the actual cell lines. So when you want to go and test that in vitro, you just pull off the cell lines with the right level. So you can pick from prostate cancer, a prostate line that has high and has low. And you're not spending the time looking at every prostate cancer cell line you can get in the lab. You're just looking at the ones you need. And if you're purchasing them from ETCC at $500 a pop, that's now a $1,000 experiment rather than a $10,000 experiment. So I suggest that all of you start using this. Another reason to start using this database is that if you publish a paper or try to publish a paper based on in vitro data using cell lines, and let's say it was the tumor suppressor P53, and one of your cell lines has a copy number variation or mutation in P53, you can no longer say that you didn't know. That data is out there. That will be used against you during publication to actually kick your paper out and say that it's fundamentally flawed. You could be writing a grant. Someone can look up your cell lines in the grant, say that pathway is aberrant in that cell line. This grant is fundamentally flawed. So you can no longer ignore this data. If your cell lines are in this database, you need to be looking at it. Once you've got a gene set, there are a lot of other things you can do with that data set. You can plug it into programs or web services like David, and what this does is lets you systematically integrate the analysis of these large data sets with what's known about their biological function. And typically the way it would work is you click Start Analysis, enter your gene list, select input type, gene symbol. You can convert between different types of gene symbols. So for those of you who do Affymetrix array data, you can actually take the probe identities, put them in here, and it'll convert it to a gene list for you, which in our lab we find very useful because as you're using data from the geo data sets at NCBI, it's often probe names rather than the actual gene names that would be given. You then are able to click Submit, and this is the typical output, so it'll give you a clustering of pathways, protein interactions, and tissue expression, for example. You can then click on pathways, and you get these things called GEO and then some kind of term. And what that stands for is gene ontology. And when I was in Edinburgh, the Gene Ontology Consortium was just getting together to come up with guidelines for annotating genomes. And without gene ontology, you actually wouldn't be able to compare between organisms or compare sequences between genes because you wouldn't have a common language to do that in. So what this enables you to do now is cluster, when you search for our favorite kinase here, PRB4, you can see that you're getting Go terms telling you that for function, there's RNA processing. You're getting that it's involved in protein amino acid phosphorylation. And so, again, I didn't need to do all the comparative genomics and everything else. Nowadays, with well-annotated human genome, you can put in that so-called unknown hit that you might have in your gene list and get an idea of its function, and then cluster genes based on their function, and start seeing whether what's happening now is sometimes it's less interesting in a disease for one protein because there might be 5 proteins with the same output in that pathway, and that any of the 5 proteins may contribute to a disease through that pathway. So you don't talk about single genes as much anymore, particularly with respect to cancer, because it's not a single gene disease, but really the pathways. And this kind of analysis lets you get at the pathways rather than just looking at a single gene. So it's the difference between finding a tree and being lost in the forest. You really want to see that clustering and the systems that come out of that data. For visualizing data, human beings have a really difficult time, for example, remembering more than 7 numbers. Lists and tables don't help you very much, but we're very visual. So if you can organize them in these kind of interesting, I almost think like tin cans on a string, but instead of transmitting information along the line, it's actually telling you how they're connected. Like, is it a protein-protein interaction? Is it co-gene expression in a cell? Or is it a common disease pathway? And this particular database, String, enables you to look at this. But as I pointed out initially, there are better tools in String, which I'm going to talk about later. String relies really heavily on automatic data mining from abstracts. And so quite often you get the wrong protein name in here because it's actually a different name in an abstract from a different organism, and their algorithms don't necessarily distinguish. And we can't afford human annotation. Okay, it was tried in Ontario for a while. There was a project called SecHound, and it was completely shut down, and its funding was not renewed because it was too expensive. So paying PhD scientists to annotate the genome is not economically viable, even though that data set today is still used as one of the gold standard types of data sets. So human annotated data is great, but it's too expensive. So we're stuck with this data tsunami, having it annotated automatically, which is inferior, but it's still better than nothing. Okay, so how are our genes and our gene lists related to each other? You can see whether they have similar sequences in their promoters, so similar transcription factors binding them. So that would connect them to a pathway, for example. A better tool than Dellaire is Opossum, and we use that in our laboratory. It has a little bit more user-friendly readout, and that database can be used to find common genes that might be regulated by a single transcription factor. So what we found, for example, in RNA interference experiments is you knock down a single protein. You get some kind of output gene expression, but it doesn't mean it's direct. It could be indirect. And so using this kind of analysis, you find transcription factors, which might be the main target that's the direct interaction of knocking down that transcript, and then it will affect 10 other genes because it's a transcription factor for those genes. So you would expect those to cluster in your gene set. You can also look at the other end of your gene, so at the 3' UTR and the exons that are encoded there. And they might be targets of common microRNAs. So this is also something you can't ignore. You can no longer put your head in the sand and say microRNAs are not affecting your pathway. Virtually every pathway has multiple microRNAs that are affecting it. And in fact, the knowledge that there are microRNAs makes it very obvious that putting a short hairpin RNA or an siRNA in a cell has to impact every single pathway that uses dicer, the enzyme that uses that to, which by the way is a tumor suppressor. You're going to overcome dicer by putting tons of this RNA into the cell. It seems logical then that all these microRNA pathways are going to be affected. So even though other things aren't affected, every time you put siRNA into a cell, you're going to affect that. And so there are new genome engineering technologies coming, and I'll just give it a little advertisement that Jamie Salzman and Jordan Pinder, who's not in the room today, two of my postdocs, are going to be giving a talk for the Cancer Research Training Program, talking about CRISPR, which is a new type of RNA-guided endonuclease for genome engineering, which lets you knock out genes in somatic cells. And that is probably ultimately going to be a much better system than shRNA libraries. The other thing with microRNAs is the databases are notorious for going out of date. So look for a really recent release date and use tools that are using new data. Okay, because the annotation, the older ones are really poor, and the algorithms for choosing the seed sequences are evolving. So you really want to be using a better, so rather than the Sloan Kettering mRNA finder, which I think is completely defunct now, you might want to use TargetScan instead, which here we can see has a June 2012 release date. You can also look at RNA binding proteins, and for a lot of you that might be a little bit far afield for you, but it's important, for example, people working in inflammation, because many of the proteins that are upregulated by interferons actually bind these purine-rich regions in your transcript and regulate their stability. So it's something you can't ignore if you're an immunologist in particular. So we're going to change gears one more time, and this is the final stretch. We're going to talk about how we use all these databases and other databases. So to look at our studies of pediatric T-cell leukemia. So how are we going to combine, again, our knowledge of computers and databases to dealing with this rather risky type of leukemia? So ALL and all its flavors is the most common malignancy in children. TLL is a common variant, and about 75% of those children will have event-free survival, and that usually involves giving them steroids and induction therapy, and their disease will resolve. But what's horrible in this disease is that if they do relapse, the 5-year survival drops all the way down to 30%. And it's for that subpopulation we really want to stratify them with respect to drug response. So how do we improve their survival? Do we develop better therapies? A lot of these therapies are very toxic. Do we stratify patients better and look for drug sensitivities? And this latter one is what I'm going to focus on. So there are targeted drugs available, but I think we can use them better because those targeted drugs do have associated toxicities. And you want to get the most benefit with the least toxicity if possible. So back to the laptop. So we're going to do some searches. First thing, we'll hit PubMed, and what do we get? We get a couple of papers. They're telling us that Notch and PTEN pathway are important. And it turns out that Notch is responsible for 60% of all TLL as a major driver mutation, and that PTEN inactivation or activation of AKT is 20% of TLLs. So that covers 80%. That's pretty good. So if you're going to focus on some pathways, focus on an important one that has the biggest impact on patients. Then once that's handled, you can start going down the list and going after the more minor pathways. What you'll find out though, and what we've learned, and I'll come to that at the end, is that the same broad pathway, even the minor variants that are showing up as drivers, still fit into the same pathway. So then you can use common drugs that might hit the linchpin or the commonality in that pathway or the last point of output, whether it's a transcription factor or a particular kinase, that might actually be able to be a great drug target for many different mutations all found in the pathway. And so we're going to explore the Notch and PTEN pathways a little bit. So what treatments are available? One thing I like to do, and I was actually introduced to this by Dr. Berman, is to troll the clinical trials database at NIH. So any NIH registered clinical trial will be in this database. They're really up to date. I mean, I think they're updated every week or every month, so it's good to check. Especially if you think you've invented something, you might want to go and check and see if it's actually, this might be part of your search to say that it's important, that you want to look at that pathway or that drug. You can type in TLL, for example. In this case, we get 346 studies. Some of these are going to be just general leukemias, in which TLL is just linked in because it's a rare type of leukemia within all ALL. And you can find different drugs, and from that, what we found is that patients are obviously treated with steroids, as I mentioned, which is the primary therapy, cytotoxic drugs, and more recently targeted therapies, and these include what are called gamma secretase inhibitors. And I'm going to explain exactly what that is in a second. But you can give these inhibitors to children with mutations in the Notch pathway, and they work, but 50% of these TLLs are GSI resistant to start off with. And for those that aren't, if you're giving it high dose, there's actually pretty serious gastrointestinal toxicity. And so this is not used as a single therapy alone. And in fact, I think they're going to start combining this with steroids to reduce that gastrointestinal toxicity. But even if you did that, it's hard to predict up front whether that child is going to respond because you already have a 50% non-response rate. So our conclusion is that we need new targeted therapies, or we need to better apply those targeted therapies to the right patient population. And so how do we find this chink in TLL's armor? Well, it would be nice to know more about the general pathway. So there's some tools for that. So if you go to Kyoto Encyclopedia of Genes and Genomes, you can type in Notch. That pathway will then come up. You can say, oh, okay, here's Notch. Here's gamma secretase. Oh, that makes sense. So gamma secretase actually cleaves off the intracellular domain of Notch, so it can enter the nucleus and act as a transcription factor with a number of other partners. And so you can understand then that if you inhibit this with compounds such as compound E, which is a gamma secretase inhibitor, you don't get the intracellular domain made, and then that doesn't turn on gene expression in that pathway. So that makes sense. That's one of those lynch points that you can target. What about its interactome? Can we learn a little bit more about that? Well, there are these great tools like Cytoscape and GeneMania that allow you to put it in gene lists. So this is a standalone Java software that's cross-platform. You can just install on your own computer. It has plugins for GeneMania, but in fact, I just use the web service for GeneMania. It's actually really great. You can just put your gene list in. It's exceptionally good for gene lists that are small in number, like 25 to 50 genes, because it kind of acts like Google. It's anticipating what you want to see rather than giving you everything, right? Because with so much data, that gets out of hand. So if you've got a really large data set, it makes it cumbersome to use. But if you have a small set, like let's say your statistical significant cutoff says there are only 10 genes that are important, putting in a 10 gene list is actually a great way to use GeneMania. So it's maybe hard to see, but when we put in Notch and PTEN, we start pulling out other factors like PES1, like AKT, like mTOR. Now we're building up some other proteins in a pathway, and if we draw a cartoon of that, it looks like this, and we're going to keep showing this cartoon as we add information to it, as we learn from using the databases, doing some experiments, and coming back and refining our hypothesis of how this pathway works and how we can target it with different drugs. So Notch fits in here because it actually inhibits one of the enzymes in this pathway through a transcriptional repressor called HES1. So Notch activation produces more HES1. That turns off P10 expression, you get less P10. How this drives T-cell leukemia is that you have growth factors like EGFR, a receptor, and it receives growth factor input. You get activation of PI3 kinase, which converts a lipid PIP2 to PIP3. And P10 actually reverses this process. And that's important because the accumulation of PIP3 is what drives activation of AKT, which activates mTOR, which then regulates the translation of a number of very important proteins and genes that are involved in cell cycle progression. So you're driving the proliferation of these cells by activating mTOR. And so you can see where you can maybe think I could target PI3 kinase, I could target AKT, mTOR, or Notch, and you get the same output. Okay? And when you develop resistance, it's because there's a short circuit in the pathway. Okay? So in the end, even if you had resistance to Notch inhibition, inhibition of mTOR may still work. And I'm going to show you some examples of that. So if you want to search for particular drugs, I like to go to PubChem compound, which again is a resource from NCBI. You can type in your favorite gene target, and here we see that you get mTOR, which is an inhibitor for mTOR is inhibited by rapamycin. So we know we can use rapamycin to inhibit mTOR. If we continue that in an iterative process, putting in each protein in our pathway and looking for inhibitors, we found that you could use compound E, which is a gamma secretase inhibitor, to inhibit Notch. We can use tricerebin to inhibit AKT, and we can use rapamycin to inhibit mTOR. Back to the lab bench. So we've done our work on the computer, we've got a hypothesis, we've got our pathway, we've got our drugs, we know our cell lines contain the pathways we need to look at because we looked at that database from Broad. I'm going to keep saying that, you guys have to use that database. Now we're going to take it to the fish, okay? And this is where I need a clinician's help, and we're going to work with Dr. Jason Berman and his group. And we take GERCAT, which are a commonly used T-cell leukemia cell line, that's very well annotated for mutations in all these genes in the pathway I'm interested in. So we look at DMSO alone, which is vehicle. We look at compound E, or tricerebin, or rapamycin. And we see of all of these, the two that work quite well are rapamycin and compound E. Okay, so that was mTOR, and that was Notch pathway. But AKT inhibition didn't work terribly well. And we said, well, okay, maybe that's just the fish. Right? The hypothesis is maybe the fish are just not metabolizing tricerebin properly. Well, what does it say in the literature? Back to the laptop. Turns out that preclinical testing for the same inhibitor against T-cell leukemia, GERCAT's actually resistant. Perfect. That means that our fish model is actually working. We have more confidence that it's not just a strange metabolite or metabolism difference in the fish that why we're not getting a response. Excellent. So now where do we go from here? We want to take this and translate it to patient care. How do we do that? Well, you can't really do that with cancer cell lines. So we had this brilliant idea. What if we just put the patient sample straight into the fish? Would that work? Could we try that? So that's exactly what we embarked on. We have our clinical protocol when a child is diagnosed in Nova Scotia with TLL, and Jason happens to be a treating physician. We get patient consent, diagnostic bone marrow sample. We do the cytogenetics on part of that sample, and part of the sample comes to the lab for xenotransplantation. What that looks like, and this work is done by Victoria Bentley and Dale Corcoran in the lab, is that the samples are collected on day 0. Density separation is used to separate out the red blood cells, for example, from the white blood cells. You can short-term culture them, but not more than 24 hours. So you can't really do drug screening with TLL samples. They're going to die. So we either inject them in fish or we freeze them down, and then we can use those in the future. And then over time, we'll actually also have clinical outcome for the child, which we would be able to use to inform hypotheses on how our drug screens were going. We had 3 patients in the last 24 months, a patient AS, EA, and TS. We had no mutations in any of the pathways that we talked about in TS, but we'll talk about some mutations in AS and EA that we found. But before we decided to do any of the sequencing, and we actually looked at what their drug response was. And I think this is the power of this approach. So here's patient EA's leukemia. We can see it can engraft in the fish. I mean, that was wonderful by itself, that it would actually engraft. We then had a problem where Normally with a cell line, we would just use a lipophilic dye to label the cell, and we would get about 7 generations within the animal where we could still detect it by microscopy. It turns out that the patient TLL samples are so aggressive that they outproliferate the label. So you can use human-specific markers like CD45 phosphatase, which is the antibodies are only going to work on human cells, and then count those even in a pool of fish cells. Okay, so we were able to dissociate the fish and do quantitative analyses as well. And so if we take patient EA, put their cancer in the fish, here we have just treatment with DMSO as vehicle, we get a tumor mass. If we treat them with rapamycin, we get a tumor mass, but it's not as large. When we gave them compound E, no tumor mass. Compound E works. Excellent. So what does that mean for our pathway? Well, it means that possibly Notch is mutated. That would be a hypothesis that we can test. So to test that hypothesis, we asked Karen Bedard's group, and in particular Marissa LeBlanc, to sequence these genes for us. So because we know AKT and PTEN are also in the pathway, let's look at them anyway and confirm that they're wild type or they're mutant. And let's look at Notch. And lo and behold, in patient AS and EA was the responder, remember, that we saw in that experiment, they actually had a mutation in Notch. Well, what does that mean? You can have a mutation anywhere. Is that actually pathogenic? Well, in the case of AS, it's a leucine to a proline. Anytime you change any amino acid to a proline, it kinks the structure. So that's almost certainly going to be pathogenic or affect the function of the protein. This one's a little more subtle, but it's an alanine which is not charged to an aspartic acid which has charged. Usually that's not very good either for protein-protein interactions. So both of them smelled like they would be interesting mutations. This is what the actual mutation in AS looked like in the raw Sanger reads. So if you've got a heterozygous SNP or a single nucleotide change, it'll show up as a double read. And so it's beautiful. You can see that we have a T to C change, and that's shown in one allele and not both alleles. Where are those mutations? Well, knowing a little bit from the databases, we have the structure of the Notch protein product. And there's this important region called the heterodimerization or hinge domain. And it turns out that both those mutations are there. And mutations in that region are known to affect the ability of gamma secretase to cleave. So they could enhance the ability of the cleavage to occur, which effectively activates Notch. So now we had an idea, well, maybe these are activating mutations in Notch. So again, we can go to databases, we go to the catalog of somatic mutations in cancer or cosmic. So whenever you get a hit and you want to find out whether it's possibly pathogenic, you need to search whether it's already been found. So it turned out the mutation in patient AS was already a known mutation that was published in Blood. So that's good, that makes sense. We're finding a mutation that's already found in a T-cell leukemia. That was not the case for this mutation in patient EA. So we decided, well, if it's gain of function, let's test if it's a gain of function. So Victoria Bentley and my group, we got these constructs from Andy Wang in British Columbia, where we have luciferase enzyme from Firefly that, given the appropriate substrate, will actually produce light. And that can be read out with a plate reader. And so what happens is if you have that intracellular domain cleaved more efficiently, you're going to get more activation of the luciferase. That creates more light. And what that looks like is that when you just take this vector with no Notch transfected in, you get a factor of 1. It goes up to 5 when you put Notch in. Okay, so that's just the basal level of putting wild-type Notch in. If you put in a delta PES mutant, and delta PES mutant is a sequence that's removed that actually is responsible for degrading the Notch receptor, you get an accumulation over time of this Notch protein product, and that leads to activation. So you get a value of around 13-fold increase. And then if you took our putative gain of function, you get an increase of around 8-fold. So it looks like it is a gain of function mutation. So we made a hypothesis based on database searches and from what we knew in the literature, and then we tested that in the lab. And again, this should be an iterative process. As you keep finding things, you go back and you check them in the databases, you form a hypothesis, you come back and check it again. Turns out that if you read in the literature that this type of mutation occurs in fully 20% of all TLL. So that's good. That's a fairly large population of patients that you can be treating. That's 20% of all of them. And these other guys that we're showing, a lot of them are all, again, in the Notch pathway. So PES mutations that affect the turnover. There's an E3 ubiquitin ligase that regulates the turnover Notch. That's also a common mutation. And so I'll just end with 3 take-home messages. The first of which is, there really are a phenomenal number of free resources, tools, and databases available for analysis of genomic and gene expression data, use them. There's no excuse not to use them. It's to your detriment not to look at these databases. One cannot live by data alone. For the bioinformaticians, I'm going to say that. And for us experimentalists, you can't do pure experimentation anymore. This really is an iterative process where you're going back and forth from the laptop to the bench, back to the laptop again. And for bioinformaticians, find a basic scientist and vice versa. You don't need to go as far as I did and married one. My wife's a bioinformatician. But really, this is an essential and potentially very productive relationship. And so I encourage all of you. And these kind of meetings, like today, are a perfect opportunity for people who are on the informatics side or the ethics side, or various other flavors of research that impact on human biology and genomics to interact and talk with each other. And with that, I'll just finish and acknowledge the work that was undergoing in the lab. So I talked about Dale Corcoran and Victoria Bentley. Our resident next generation web biologist using informatics is Jamie Salzman. Great work from the Bedard lab and all the sequencing projects, of course, Jason Berman's lab. And then our other collaborators, Andy Wing providing cell lines and samples for the luciferase assay, and Dr. Lenzon, who provides the castor fish, funded by NSERC and CHR through the CHIRP program and also through the Open Operating Grants program. And I also have to acknowledge I'm a senior scientist at Beatrice Hunter Cancer Research Institute. Thank you, and see you for the
Thank you, Brian, for a great talk. Anybody have any questions?
I mean, they can be philosophical. I think my talk was kind of that bent.
Andrew?
So, well, comment and question. First comment is that in your construct there of laptop and bioinformatician bench research, I think the fact that Jason wears two hats, a clinician and a researcher, is probably...
That's the next talk.
So, really, I think it's a complete bench, bedside laptop, and that cycle is probably more helpful. As we're lucky here to have all those resources. The other comment or the question I would have is, what sort of, given that you have postdocs and masters, other level students, what sort of resources do you have to do sort of basic bioinformatics training with respect to, for example, open access databases as opposed to active manipulation of large data sets?
So this is kind of a work in progress. Myself and Dr. Salzman. So Dr. Salzman, for a little background, trained at Dell with Roy Duncan, went off to Toronto to the Big Smoke, got some additional experience there, and then came back to my laboratory. And he's a really great teacher. He's also a web biologist, and now he's heavily getting into informatics with our resident bioinformatician, Dan Gaston, helping along the way. And we've been talking about having workshops. This is something that we want to integrate. So he's a former cancer research program trainee, and to get back to that program for supporting him and being such a great program, we're going to try to mount some integrated learning sessions talking about this type of technology. There are programs, travel awards available on the cancer side where you can send people away to learn this. There's also free travel awards for the Canadian Bioinformatics Network. And Dan Gaston and Jamie and myself have been talking about hosting, potentially with Morgan Langell now, a new faculty member in pharmacology, having workshops held right here in Nova Scotia or in the Maritimes for helping students learn about these things. So I benefit being in the UK that I went on Sanger Center workshops. So you're absolutely correct that workshops do help facilitate this kind of thinking and using these tools in your research. In the meantime, you can always check my website. We have a lot of these links on our website. And in fact, Jamie has also talked about putting in a kind of more specific genomics set of links in our website material. So if you type in Dellaire Lab in Google, you can pull that up, and that might help get you started. I can also provide links for some of the databases we talked about today, if people find that useful. And Sue can circulate those to the mailing list of attendees. Chris?
Graham, when you're looking at databases, it's like with a lot of data, it's garbage in, garbage out. So how do you ensure that the database you're using, or at least try and be pragmatic about which databases you should be using, because they come and go so often in terms of which ones are useful or which ones are useless?
That's a moving target. So we have colleagues at Stanford, Stephen Montgomery in pathology at Stanford University, and we use him as a sounding board. We have a colleague Ryan Morin at Simon Fraser, and we bounce these things off these people and say, we're using this, does that make sense? And even the pipelines that are used for analysis, there's no standardized pipelines. Almost every laboratory has its own process. And through trial and error, people are sharing this. I know they're trying to develop standards, but it's hard to develop standards around databases when their shelf life is 5 years. You know, it's a lot of investment that's gone. So that's, in Canada, we can be doing more, but there's no political will or money behind having an equivalent of NCBI if we use NCBI as a resource. So, you know, eventually things filter down and end up in NCBI in one form or another. I would say that's probably your most trusted source, that and Sanger, for example. Other kind of lab-centric databases use your own risk. And that's where the biologist comes in hand. Another point I wanted to make, but I thought 3 points were enough for our brains to remember at the end for take-home messages. But a fourth point, if I could add that, is that for the bioinformaticians, your cutoffs for significance are totally arbitrary. They don't always have any biological relevance at all. So show your data. The best thing to do is just give them a rank-ordered list and just let them look at the data. We pulled out hits that were not significant in the analysis that was done, that we go and test with an antibody and do a Western blot, and it turns out to be significant. We found a splicing change in one project that when the statistical analysis was done was irrelevant, but in the lab, there's no protein, even though that splice event was such a minor splice event that was seen in the data set. So you really have to balance biology. This is, again, where it's a perfect marriage, and if it's clinical relevance, you'd have a clinician also informing you that that's a good question, that's clinically relevant. Sometimes we get caught up in the minutiae. And so I think having that input is good as well. But there's no substitution for experience, reading the literature, and interpreting it from the biological side. Okay. Thank you all.